So Mendel now had to come up with an explanation for why was it that the white flower color disappears entirely in the F1 generation hybrids, and then why it comes back in the F2 generation, and why does he get that interesting 3-to-1 purple to white flower ratio. And so this brings us to a discussion of Mendel's model. So as we discuss Mendel's model now, a couple things to note. There are four different sort of concepts that are all very much interrelated that make up his model. And when Mendel was coming up with his model, he had no idea that chromosomes and genes existed. And so he dubbed this term discrete heritable units, which we actually now know are genes that are found on chromosomes. And so what we're going to do is we're going to go through Mendel's model, and instead of thinking of it in this abstract discrete heritable unit way, we're actually going to apply what we now know and understand about chromosomes and genes and all that good stuff. So sort of superimpose that on his model to get the full picture as we understand it as modern day geneticists. Okay, so first of all, Mendel reasoned that only the purple flower factor was somehow affecting flower color in the F1 hybrids. And this is why they all came out purple, and there were no whites, right? So it was only this purple flower factor that was affecting the character in these in the F1 generation hybrids. And so Mendel called this purple flower color the dominant trait for the character, and the white flower color a recessive trait. All right, so the dominant trait appears in that particular character, and then the recessive is kind of hiding in the backgrounds, and it has no contribution to the actual appearance of that flower. And he also noted that the flower color, the white flower factor, it wasn't diluted in the F1 generation hybrids. It wasn't destroyed because it had the chance to reappear when he crossed the F1 hybrids to themselves in the F2 generation, right? He got that 3-to-1 ratio of purple to white flowers. That white flower had a chance to come back in the next generation. All right, and Mendel was a good scientist. He didn't just do this once and said, oh, hey, look at this new result that I got. He repeated these experiments over and over and over again, and he used not just flower color, but he used many other characters in these P planes as well, and he always got the same result. So if we take a look in seed color, his experiments there, seed shape, pod color, where the flowers are located on the plant, stem length, he always got just about a 3-to-1 ratio of the dominant trait, 3 of the dominant trait, and only 1 of the recessive in that F2 generation. So he said, there's got to be something here. I have to be able to explain this now. And again, what Mendel called this heritable factor or discrete heritable unit is what we now know and understand to be a gene. So we're going to take this information and apply it now to Mendel's actual model. So his model is essentially a hypothesis to explain that 3-to-1 inheritance pattern that he observed in the F2 offspring. And again, there are 4 interrelated concepts that make up this model. And we're going to talk about them in the context of what we now understand as in terms of genes and chromosomes. So here's the first concept. First concept states that there are alternative versions of the genes that account for these variations in inherited characters. Example. So we're looking at flower color in the pea plants. Flower color exists in 2 versions. 1 for purple and 1 for white. And so he thought that there was this gene or discrete heritable unit for each of the 2 versions. 1 gene for purple flowers and 1 gene for white flowers. And when we have a gene that exists in these different versions, we call these different versions of a gene alleles. We're going to be using this term a lot. So an allele is a different version of the same gene. Now, if we look at this first concept and apply it to the genetics of the F1 hybrids, let's take a couple steps back just to refresh our minds what these F1 hybrids are. These are the middle generation. They are the hybrids that result when you cross one kind of flower to a different kind of flower. Both of these parental flowers, remember, being true breeders. And so in this case, this purple flower inherited one set of chromosomes from the purple flower and inherited one set of chromosomes from the white flower, right, to make this diploid flower. So if we go back now, here we've got one chromosome from the dad in blue and one chromosome in red here from the mom. One of these chromosomes from one set and one from the other set. And this particular chromosome in each set happens to hold the gene at this particular locus here, the gene for flower color. So you'll notice this chromosome, the blue one, came from the purple flower because it holds the allele, the purple allele for flower color. And this chromosome came from the white flower because it holds the white allele, or the white version of the color gene. If we take a closer look now, this gene, the purple gene, if we look at the sequence of A's, T's, C's, and G's for this gene, you'll notice that at this first position here, there happens to be a CG pair. And this sequence of nucleotides will code for the production of a protein, which is an enzyme which has the ability to make purple pigment, making this flower purple. The white version of the gene, if we look at its sequence, it's a little bit different. At this first position that's shown here, instead of having a GC pair, there's an AT pair. And what this does is it prevents the production of a protein. So in this case, no enzyme is made from this particular gene. And if no enzyme is made, no enzyme can be used to make more purple protein, or more purple pigment for the flower. But in this case, we're looking at the F1 generation hybrids, right? So they got one purple gene and one white gene. Having that one purple gene or one purple allele of that flower color gene is perfectly enough to make enough of that enzyme to make that flower appear purple with the amount of purple pigment that's produced. Okay, that's a lot of information. Let's go back and review, right? So the first concept in Mendel's model is that alternative versions of genes account for variations in inherited characters, meaning there's a purple version of the flower color gene and there's a white version of the flower color gene. All right, second concept, and we've kind of looked at this already on the previous slide, but for each character, an organism is going to inherit two alleles, one from each parent. This makes a lot of sense, right? Because you get one set of chromosomes from your mom, one set of chromosomes from your dad. So you have two copies of every gene. Now, sometimes you might inherit the same copy of a gene for a particular character. If this happens, if you have two identical alleles, one from each parent at a particular locus, you are called what is homozygous for that particular gene. So an organism that has two identical genes for a gene is called, two identical alleles for a gene is called homozygous. And this is definitely the case for those true breeding parental generation pea plants that Mendel used as his very first cross. They were both either homozygous purple or homozygous white. On the other hand, you don't have to inherit the same allele from both of your parents. You might inherit one version of the gene from one parent and another version from the other parent. In this case, if you are an organism that has two different alleles for a particular gene, you are what is called heterozygous for that particular gene. And this is the case for the F1 hybrids. Right? If we take one step back, right, they inherited one purple allele from the purple true breeding parent and one white allele from the white true breeding parent. And so they are heterozygotes. The F1 generation are heterozygotes for the flower colored gene. All right, third concept. If you are a heterozygote, meaning if the two alleles for that particular locus are different, one of them is going to be a dominant allele, and the other one is going to be recessive. The dominant allele is going to be the one that determines your appearance. And the other one, the recessive, has no noticeable effect on the appearance. So let's go back to the F1 hybrids, right? The F1 hybrids, these guys, they inherited a purple allele from the purple flower and a white allele from the white flower. Now you'll notice that these flowers came out purple, indicating that the purple allele is the dominant allele because it determined the appearance of that flower. The white allele is the recessive allele because it has no noticeable effect on the appearance of those F1 hybrids. And then the fourth concept. The fourth concept brings us to Mendel's first law of genetics, which is the law of segregation. Law of segregation states that the two alleles for a heritable character will separate or will segregate during gamete formation and end up in different gametes. So let's think about how the F2 generation were created. You take the F1 hybrids and you allow them to cross to each other. This means that they have to go through sexual reproduction. So they have to make their eggs and their sperm. Now, the F1 hybrids, when you look at their chromosomes, they've got one chromosome with the white allele and one chromosome with the purple allele. Remember that during meiosis, during gamete formation, you are separating sets of chromosomes. So the homolog with the white allele is going to go to one set of gametes, and the homolog with the purple allele is going to go to a different set of gametes. So each egg or sperm only gets one of the two alleles, either the white one or the purple one, and then can pass on that one allele to the next generation. And again, this has everything to do, this law of segregation has everything to do with the way that homologous chromosomes are sorted into different gametes during meiosis.